Hello, my name is Garrett Yount, and I'm presenting some fun and interesting results of a study on behalf of the whole team of scientists at the Institute of Noetic Sciences, including Helene Wabe, Arnaud Delorme, Lauren Carpenter, and Dean Radin. So this was a fun study that we did that was inspired by a few things. One was an anecdotal observation that happened about 20 years ago during an experiment in Beijing, China with Qigong masters. So let me just start by defining clairvoyance just for the purposes of this talk. I'm talking about the faculty of perceiving things or events beyond normal sensory contact. So the anecdote that happened those years ago, it's a long story. I'm going to keep it short because I'm going to tell a couple other stories before we get to the data. Bottom line is, in Beijing, in a hospital setting, me in a white coat brought in a Qigong master and said, hey, I want you to send Qi to cells in this Petri dish, and they're normal brain cells. We did the whole thing. We walked out and he's like, no, those are not normal. They had a lot of disturbance Qi and we need to send them away. In the end, it was a miscommunication and he was right. And one of the things that stood out to me is that I was in this white coat in a sterile environment telling him the way it was, and he got some information that he knew I was wrong. He was right. So that left a big impact on me. And I wanted to see if we could bring this into the laboratory. So we did do an experiment, and this is published. So I'm going to just talk about it because it's background and some of the design that we learned through this published study, we applied to the current study that is unpublished. So I'll run through this somewhat quickly. So to try to set up this event, but in the laboratory, the first thing we did was bring folks from our local area who were self-described clairvoyants. They operated within the community professionally in this way. Brought them into the lab and said, hey, this is what we want to do. So before designing the experiment, we brought them in to collaborate. And in this case, instead of having normal cells and cancer cells, I just wanted to make the signal as clear as possible and make it as simple as possible to replicate what we saw, which was the detection of cancer cells. So we had cancer cells in a Petri dish, and then a Petri dish that had, calling it salt water, it's basically the buffer that allows the cells to stay alive in the Petri dish. And we just set them side by side. And there, of course, you can't see them with your naked eye, but we asked them, can you tell the difference? Are you feeling any kind of subtle energy coming out of the Petri dish with the cancer cells in it compared to just the salt water? And we wanted to have 10 participants, 10 clairvoyants in this study. So as we interviewed folks, a few of them or a couple of them said, you know what, this is not the kind of thing I do. Who knows, maybe they really wanted to work with the whole body, but these little pieces of human cells in a Petri dish, they just weren't feeling it. We also then put the dishes inside of an envelope, just again to be clear that there's going to be no visual cues, and make sure that the paper doesn't interfere with the emanations of these subtle rays. And so we interviewed 12 people until we got 10 yeses. So 2 of them said, this is not the way I work. So we had 10 folks to move forward with. So the first test was bring them into the lab, put in front of them an envelope that's unmarked, and inside of it is either the Petri dish with cancer cells or the Petri dish with just the salt water. So we had 20 participants altogether. We had the 10 sensitives and then 10 staff members who you just asked them, these are basically the technicians saying, no, I don't have any clairvoyant skills necessarily. So these were our control subjects. We did 34 trials, and we chose 34 because the biostatistician on the team, who's been decades in biomedical research, we said we want to be conservative about this. If we see something here, we want the statistics to be strong. So he said, well, it's just like a yes or no choice. So you need to do it 34 times for it to be, you have enough power. So we did that with all 20 people. The results came out of chance for everybody. So I was crestfallen, scratched my head, brought in a few of the practitioners, said, hey, help us think about this some more, because I know this can happen. Why isn't it happening here? So the first thing they said was that, during the orientation, when you brought us in, there was something to compare. We knew there were two things and there was something to compare. And this is a really, really valid point, because if we were in person, I would demonstrate, because if you get two little paper cups of water that have slightly different amount of water in them, if you're holding them both in your hand and you can compare them, it's really easy to figure out which one is heavier, which one has more. But if you put them down, blindfold somebody and had them one of the cups and then had them the other one, it's really hard if there's nothing to compare. So we said, great, okay, let's do it all over again. And this time we'll put the unknown in the middle, and we'll put the cancer cells on the left and the salt water on the right. And that way you can compare back and forth between them and have this comparison when doing the trials. So we did that, same participants, 34 trials again. Again, everybody came out of chance. So again, scratch our heads, talk to the practitioners, like what can we change? What's going to make this able for us to reveal what we know can happen? And says, well, the thing is 34 trials. It's like really onerous. It's like boring, and it's like too much. 34 is too much. So we took that feedback. The statistician was pushing back because there was no evidence that the accuracy waned over time. But it was regardless, the whole thing is just feeling too onerous. So I was like, how do we make it less trials? 10 trials, we just want to do 10 trials. So it's like, all right, this is how you do it. You put 10 envelopes in front of them at the same time. One of them has the cancer cells. 9 of them have the salt water. And then you just pick which one out of the 10. And somehow the numbers compute in a way that 10 trials gives us enough power. So we're like, great, here we go. We did it again. Same 10 subtle energy perceiving clairvoyance, 10 staff, only 10 trials this time, and everybody got a chance again. So not conclusive, but definitely not, being honest, not what I wanted to see, but we'd run out of money. We published it. So this is all the background. So this has still been like this yearning I've had, like, okay, I wanted to see this happen in the lab, wasn't able to do it before. And then this coincidence happened at our research institute. We had the honor of hosting a Buddha relic at our research institute for a short time last year, actually 2 years ago now. So for those of you who don't know, a Buddha relic is a crystal-like object that is purportedly found among the cremated ashes of Buddhist spiritual masters. And these are treated with very much reverence, and they're commonly believed to emanate a subtle energy that is perceptible to people that are sensitive to that type of thing. So we had this with us. So we thought this would be an awesome opportunity to try to do an experiment like this again around perception of subtle energies. So we wanted to set this up basically to test whether without visual cues, this clairvoyance could detect it. So we recruited professional clairvoyants, again, from the community. So we didn't do any, we basically trusted this is they're professional. They work in the community, and they have excellent references. So this is the population of folks that we wanted to work with. So we ended up bringing in 4 of them, 4 self-identified clairvoyants. And again, they provide these services in the community. And then we recruited 3 of the technicians again to serve as our controls. And again, they felt that they didn't necessarily have any special skills to see subtle energies. So this is a smaller sample this time. We had 4 clairvoyants and 3 control subjects. Of course, we had blinding applied to all of the staff. And again, prior to the start of the test, the research assistant would place the relic in an opaque container in the center of a table inside. In this case, we had it inside an electromagnetically shielded room. And again, we had this orientation phase. where each subject, both the controls and the clairvoyants, were able to orient around it. So as you can see here, the little urn in the front has the relic inside of it. Now these are the 10 lined up behind it are going to be used for the experiment. But the first stage was to bring the subjects in and give them about 5 minutes to orient and feel what they can feel from the urn that has the relic inside of it. And again, it's about 5 minutes. Have them, and I was with them. So during the orientation and the test, they're allowed to have their hand just as you see here. No touching it, but within about 1 or 2 inches, like really, really getting close to it and getting intimate, but without touching. So after 5 minutes, I escorted them out of the shielded room and out of the building into another building. So there's no way for an indication, a crossing of visual cues with the staff that then went in and moved the relic from the front urn. Sorry, I'm trying to point to this, to one of the 10 urns in the back that are numbered. And this was determined by a random number generator, which urn it was to be moved to. And the RA would lift the lid of all the containers in an attempt to equalize the amount of touching and manipulation of each of the urns. So then the RA goes out of the room and out of sight, and then I bring the subject back into the building, back into the shielded room, and sit there quietly and watch while the subjects put their hands over all 10 of the urns to try to detect which urn has the relic in it. Again, no touching. They were given, between 5 and 10 minutes, and then recorded the number of which urn they believed it to be in. Then we left the room, and we basically repeated this process 10 times. So as you remember from the published report, if we've got 10 choices, then picking 1 out of 10, we only need to do it 10 times to have enough power. So now we want to move to the results. So it's quite a simple experiment. And in this case, we only did it 1 time. So we're getting to the culmination here. So the first result that I'd like to describe is that during the orientation period, this is when I escorted in both the clairvoyants and the control subjects to spend 5 minutes in this quiet room with the relic inside of the little urn. This was a period of getting, discerning if what getting used to what they see, since there has never been any visual contact with the relic. So all of the seers did describe a variety of sensations and perceptions associated with the urn that had the relic in it, as compared to the 10 empty urns in the back. And none of the control subjects reported such perceptions. So this seemed to be as expected. Now for the experimental trials, what we found again was that all of the subjects found we had chance. Everybody was at chance. These are the averages for the 2 groups averaged with standard deviation. So you can see there was quite a bit of variation, but there was no statistical significance to the results of either of the groups. So must admit, wasn't super happy about these results, but data are data, right? So these are the data we found. So our conclusion from this small trial was that we found no evidence that clairvoyant seers were able to detect the presence of the Buddha relic without visual cues. So in terms of discussion, I've got a couple of thoughts I'll share, and then I hope that during the Q&A, we can get a bunch of other perspectives on this. I think the first thing I would say is that it's quite possible that the subtle energy emanations from the relic, we don't know how far in space and even what kind of space they emanate in, but it's possible that they would radiate far enough. This is about a 2-foot space in terms of where the empty containers are and where the one that was containing the relic. So it's possible that the emanations are just too strong, they go too far to be able to focus in and see without interference of where it is compared to just a little bit further away. So that's one possibility. Another possibility is that there are some traces left in the containers and it contaminates it and it becomes all complicated. Again, we didn't see any indication of this in terms of the accuracy falling over time because, during the first trial, it would be less so. And then, of course, these are the same containers used across all of the subjects. So lastly, I'd like to say that in terms of the urns being empty, the empty space inside of the urns, I would expect that most of the crowd here would agree that in terms of energetic emanations or subtle energies, empty space is, probably anything but empty. And it wouldn't be surprising that there are some kind of signals that could be perceived from empty space. So it does present, I think, something to think about for the design of future studies. Because it looks like what we see from these data, it looks like a signal to noise ratio may be an issue. So there may be signals coming from the Buddha relic, there may be signals coming from empty space. And in terms of being able to distinguish those, that's why I'm calling it a signal to noise ratio issue. I think that would be an important design element to think about for future studies. So I'll end now, so there's plenty of time for Q&A. I want to thank all the folks that helped carry this out. Again, the scientists on the team, as well as our staff, Joyce Anastasia, Meredith Pearson, Heather Mendel, Sue Steele, Dee Mers, Stephen Swanson, Cedric Kennard, all the clairvoyant seers, and the entire staff at IONS. And most importantly, I want to thank Emerald Gate, our funder, and acknowledge their wisdom and their mission to fund research like this. I really very much appreciate it. So thank you.